Hello, everyone. I'm Courtney Mobilian, and I'm from Indiana University, and I will be talking about the effects of sea level rise and saltwater intrusion on a tidal freshwater marsh on the Altamaha River, Georgia, and the experimental approach that we took to look at this issue. So tidal freshwater marshes are threatened by climate change, which includes things like rising sea level, decreased freshwater input from rivers, increased drought, and more frequent and intense storm surges. So with climate change, these tidal freshwater systems are increasingly vulnerable to saltwater intrusion. So we wanted to look at different disturbance types of salinity. So we wanted to see how press or continuous saltwater intrusion versus press or short-term saltwater intrusion affects things like biogeochemistry, plant communities, soils, and microbial communities in a tidal freshwater marsh. So to do this, our experiment consisted of 5 different treatments. So we had a press treatment, and those plots experienced continuous high salinity, high pore water salinity of about 2 to 5 parts per thousand. And this was meant to simulate long-term or continuous sea level rise. We had a pulse treatment, and those plots were exposed to short pulses, so 2 months out of the year, short pulses of salinity several times during low flow. So low flow on the Altamaha River is typically during September and October. And so this treatment was meant to stimulate short-term drought or acute saltwater intrusion. And then we also had a freshwater treatment. And this was just to see if there were any effects of the increased inundation or extra water that we were putting on the plots. So we just wanted to see if that in itself had any effects on the marsh. And then finally, we had 2 control treatments, so one with siding and one without. And I'll show a picture of this siding in a couple slides, but the siding was just set up around the plots to help hold in that treatment water when we started dosing. And there were a total of 30 plots, so there were 6 replicates for each treatment, and each plot was 2.5 by 2.5 meters. And this experiment was called Seawater Addition Long-Term Experiment, or SALTEx for short. And this is an aerial view. So this is our experiment right here, and this is the Altamaha River right here. And this is a tidal freshwater marsh, so it does experience twice daily tidal inundations of freshwater. And this experiment, the dosing experiment, started in April 2014, and it went through December 2017. And these are our saltwater holding tanks. So we would truck salt water, which is about 30 parts per thousand salinity. We would truck that from the coast to our inland site here and store it in these saltwater holding tanks. And this is our pumping or mixing station. So this would pump fresh river water, which the river is just on the other side of these trees here. So it would pump it and mix it into those big saltwater tanks that I just showed. To create our brackish treatment water, which had a salinity of about 15 parts per thousand. And this pumping station was also used to pump the treatment water to all of the individual plots once we started the dosing. And this is what one of our plots looks like. So this is the siding that I was talking about earlier, and that was just to help hold in that treatment water to allow it to infiltrate the soil while we were dosing. And so our press treatment was dosed 4 times a week with that 15 part per thousand salinity treatment water. It was dosed with about 70 gallons 4 times a week for 3.5 years. And then our pulse treatment was dosed 4 times a week just for that 2-month dosing window in September and October. And then the rest of the year it was dosed with fresh river water. And then our freshwater plots, those were dosed the same. So they were dosed with fresh river water 4 days a week for 3.5 years. And then of course our control plots didn't receive any treatment water, but all of the plots still experienced the natural tidal inundation. So the plot siding was still low enough to allow the tidal water to infiltrate into the plots. So that didn't affect it at all. This is just what one of the fountains looks like in the plot. So this fountain actually is what delivered the treatment water to our plots. And down here is just a splash plate to help prevent erosion from when the treatment water was being applied. So first thing let me explain this graph briefly because I'll show a few of these and there's kind of a lot going on. So on the x-axis, we have time down here. And then our press treatment, like I said, started in April 2014 and went through December 2017. And so the duration of that press treatment is indicated by this light gray shaded box here. And then our pulse treatment, those were dosed September and October every year. So that duration is indicated with these darker gray shaded bars every year. And then we also did take measurements twice before we started dosing so we could get some background or baseline levels for our pore water chemistry. And then our press plots are indicated by these red circles. Orange is our pulse treatment, blue is the freshwater, and then the light and dark green are our controls. So the pore water salinity immediately increased right after we started dosing. And then within about 3 to 4 months, we reached that target salinity of about 2 to 5 parts per thousand. So salinity was really elevated in our press treatment and stayed elevated throughout that 3.5-year dosing period. And it was also elevated in the pulse treatment just during that 2-month dosing window. And you can see that it pretty quickly declined to background or baseline levels just a few months after we stopped dosing the pulse treatments. Pore water ammonium also significantly increased in the press treatment. So after about 6 months, it was really elevated in our press treatment and it stayed pretty high throughout that 3.5 years. There was also a slight bump in the pulse pore water ammonium, but it's hard to tell. It wasn't to nearly the scale as it was in our press treatment, and it wasn't sustained like it was in the press treatment. So it went back down almost immediately to baseline levels once that pulse dosing stopped. The pore water phosphate also increased. So after about 6 months of dosing, it was significantly elevated in our press treatment. And again, it stayed elevated through that 3.5 years. And we also saw a little bit of elevated phosphate in our pulse treatment as well, though not to nearly the scale as it was in our press treatment. We also looked at pore water sulfide, and that was important because that's a product of sulfate reduction. Unfortunately, we didn't start measuring sulfide until about 6 months after dosing started, but it followed a similar trend. So it was significantly elevated in our press treatment and stayed pretty much elevated throughout the dosing experiment. Didn't really see a change in pore water sulfide in our pulse treatment though. We also looked at how the vegetation responded, and not surprisingly, the salt sensitive species declined in percent cover. And we looked at 4 dominant species in our marsh. So there's Ludwigia, Polygonum, Pontederia, and Zizaniopsis. And on this figure, these dashed lines here, those indicate when dosing started. So for 3 of our 4 species, we do have preliminary or baseline data. And so for percent coverage, the Ludwigia immediately declined to 0% coverage after we started that press dosing. And it also actually disappeared to nearly 0% in our pulse treatment as well after that first October dosing. For our Polygonum, that decreased to 0% cover as well, but that took a little bit longer. So that didn't disappear until after about the first growing season. And unlike Ludwigia, Polygonum did not disappear from our pulse treatment. And then Pontederia and Zizaniopsis, those took a little bit longer to respond to the press treatment. Took about 6 months or a year to see declines in percent cover for those 2 species. And we did not see a change in percent cover in our pulse treatments for these 2. And they also did not disappear completely in the press treatment. So even after 3.5 years, there was still a little bit cover of these two species in our press plots. This is what these plots look like. So here's our control plot, and you can actually see 3 of our 4 dominant species. So we have Zizaniopsis, here's some Pontederia, and then here's some Polygonum over here. And then this is what our press plots look like after about 3 years of dosing. So you can see it's pretty bare. There's not a whole lot going on. There is still a little bit of Zizaniopsis and a little bit of Pontederia over here, but other than that, it's pretty devegetated. And these are our porewater wells that are also visible now. So that's what we used to sample our porewater from. And again, this is just the fountain that delivers the treatment water to the plot. And then this here is a base or a collar for a static flux chamber that we use to measure greenhouse gas emissions. So we also looked at carbon cycling. So we measured carbon dioxide and methane emissions using a static flux chamber, which is this right here. And so ecosystem respiration was significantly depressed in our press treatment. So after about 3 or 4 months or so, it really declined in the press treatment, and it stayed low throughout the 3.5 years. But for our pulse treatment, we didn't really see a change in respiration. Methane emissions were also depressed in the press treatments, although maybe not as much as ecosystem respiration, but still consistently lower in the press treatments, and it stayed low throughout the dosing experiment again. And there was really no change in methane emissions from our pulse treatment. We also looked at soil surface elevation change. So to do this, we used sediment erosion tables, or SETs. And the way that these work, so these have a stable benchmark that's driven into the ground outside of the plot. And then this arm is attached to that, which then extends over into our plots. And these pins here are used to take repeated measurements in the same location over time. And so this allowed us to track elevation, soil surface elevation change in our plots throughout the experiment. And so there were significant changes in soil surface elevation. So the controls and the freshwater treatments, they actually gained about 2 to 3 centimeters of elevation over 3.5 years, which is close to tracking sea level rise. And then compared to that, the press treatments, they lost almost 2 to 3 centimeters of elevation over that same time period. And this loss in elevation was really linked to decreased root biomass and decreased root productivity in these plots. And then we have our pulse treatment. There was really no change in elevation, just kind of stayed static throughout the experiment. So from that data, we were able to calculate carbon gains and losses from the soils. So as we saw, the press plots lost nearly 3 centimeters of elevation over 3.5 years, which resulted in a loss of carbon of about 770 grams of carbon per meter squared over that same time period, which if you compare that to the controls and the fresh, which gained anywhere from 250 to about 400 grams of carbon per meter squared, which is a significant difference. So this really shows that even just a small change of a few centimeters in elevation has a significant impact on carbon storage in these soils. Again, with the pulse, there was really no change in carbon gains or losses, but I mean, they're still clearly different from the rest of the treatments. Bulk density and organic carbon, we didn't see a change across treatments really for either of these. So that carbon loss in the press treatment was really driven by that elevation loss in those plots. Finally, we looked at microbial community composition. And so there are actually 3 distinct or unique microbial communities. So the press and the pulse were different from both each other and also from the controls and the freshwater treatments. And these changes in community composition were driven by changes in environmental variables, which are shown as these vectors here. So those changes were driven by things like salinity and nutrient concentrations like inorganic nitrogen and phosphorus. Sulfate reducer abundance significantly increased in the press plots and also somewhat increased in our pulse plots as well. We expected that methanogen abundance would decrease in our press plots because there have been a lot of studies that show, seawater intrusion depresses methanogen activity, but we actually did not see any change in methanogen abundance across our treatments. So that was a little bit surprising. Finally, microbial diversity was significantly reduced in our press treatment. We didn't see a change in the pulse treatment for microbial diversity, but this reduced diversity in the press treatment is evident in ecosystem functioning, which is demonstrated here. So we have carbon acquiring extracellular enzyme activity, and it was significantly reduced in our press treatment here compared to the other treatments. And then going back to ecosystem respiration again, not quite as clear cut as the extracellular enzyme data, but still it was lower in the press treatment compared to the other treatments. So to wrap up, press seawater intrusion resulted in a lot of changes in the freshwater marsh. So it resulted in a release of inorganic nitrogen and phosphorus, particularly ammonium and phosphate, resulted in decreased plant productivity and diversity, a decreased ecosystem respiration and methane emissions, increased soil subsidence, which led to significantly reduced carbon storage, and finally reduced microbial diversity and carbon cycling. And so we really think that the plants are what's important and what's controlling these systems. So the plants provide food or carbon to these systems, and when your plants disappear, your source of carbon, especially labile carbon, disappears. And then you start seeing changes in microbial communities and carbon cycling, which results in changes in your ecosystem functioning. And so with that, I would like to thank all of our students and technicians and everyone who worked on this project to make it possible. And I would also like to thank the Georgia Coastal Ecosystems, LTER, the National Science Foundation, the Department of Energy's Joint Genome Institute, who sequenced our microbial data for us, the Marine Institute at the University of Georgia, and finally the O'Neill School of Public and Environmental Affairs at Indiana University, all of who helped fund this project and make it happen. And with that, I'd like to thank everyone for watching.